So thanks for being here, and thanks for the opportunity to speak to you. I am going to talk about AGTC. We did go public, so we do have a safe harbor. We are an AAV gene therapy company. We use the AAV vector. It does not affect the germline, so it does not integrate. Rather, when you give an injection, the AAV vector goes into the cell, works its way through the cytoplasm, into the nucleus, uncoats, becomes an episome, and expresses protein for the life of that cell. It is our focus to be the world leader in gene therapy ophthalmology. We have treated over 100 patients across 3 different indications. The company has been in business for quite some time, and we really have a good understanding of the vector selection, design, and the delivery. And delivery is very important in this space. And we've brought 3 programs from inception into the clinic. From an IP perspective, we have over 100 patents, and they cover all of our products as well as our proprietary manufacturing system. We've got a broad pipeline, and we have 7 things that we're working on our pipeline, which we will talk about. And we do use next-gen vectors like the previous company, as well as some of the standard AAV vectors. We've seen safe and sustained delivery with the next-gen as well as the standard vectors. In dog models, we have over 10 years' worth of sustained expression in our Achromatopsia. And in our alpha antitrypsin program, we have over 3 years of sustained expression in humans. So AAV is a pretty simple, safe. There's never been an SAE related to clinical trials or related to the AAV vector. It's only got 2 genes, a rep gene and a cap gene, so it's very easy to manipulate. It's stable. We've seen it's resistant to a degradation, easy to purify, and you get very good long-term storage. The capacity is there. 90% of the human genes can be put into the AAV vector, and we get very good sustained expression, basically 1 dose and for the length of the cell life. So as I said, we have 7 programs. We also have an XLRS program like the previous company. Ours is a little further along, I believe. We start in data are in the clinic next year. We also have an Achromatopsia program. These are all inherited retinal diseases, primarily affecting boys, but also working to young men. We also have an XLRP program, which we'll be working on. It's about a year behind that. And then at the bottom here, we have our alpha antitrypsin deficiency program, which will go into phase 2B next year. So I should say that these are all monogenic diseases. These are all monogenic diseases. There is a good regulatory pathway, typically visual acuity, and we have good animal models, both in mice, in some cases in dogs, and in non-human primates. So XLRS, between the US and Europe, is approximately 35,000 patients. Patients are missing the RS1 protein, so you get sort of a delaminating of the retina, and that's what you see. The top picture would be the normal eye. The bottom picture where the white arrows are, as you see these kind of holes being created. Once you begin missing that structural protein, there's a significant risk for vitreal hemorrhage, and most of these patients will go blind over time. Obviously, normal things like driving, et cetera, are significantly affected. Our development pipeline is at the bottom here. You can see that we've already begun our natural history studies. We've completed our talks. Our phase 1/2 program will begin in 2015. And because this is gene therapy, we will get an initial readout 12 weeks after the trial is over. So we will actually have data in that program next year. So our second program in ophthalmology is Achromatopsia. There are approximately 27,000 patients in the US and EU. Here the patients have a lack of cone photoreceptor function. This occurs at birth, of course. These patients are typically young boys. They cannot see in the daytime. They can only see in fairly dim light. And hence you see why the boy has his eyes closed. These children typically have very dark glasses on, or even with a red tint, in order to help them once they're outside, or even if they were in, say, a normal office light in an office building, they also would still have a real problem with seeing. They also have terrible visual acuity. Their best visual acuity, aided visual acuity, is 2200, so these boys are legally blind. We have a dog model, which unfortunately does not translate well when you put it into a PDF, but what we can show you in that dog model is that untreated dogs cannot go through a maze that have the Achromatopsia. There's a nice dog model for this. Treated, the dogs can go through the maze no problem, even at increasing light intensity. So we're very optimistic for this particular program. Again, it's almost right on par with the XLRS, where we've already done the natural history, or we're working on those. The tox study is going on right now. This program will also go into the clinic in 2015, a phase 1/2, and we will have readouts by the end of the year. And then this last program, XLRP, X-linked retinitis pigmentosa, this is a little slower progression. It is a form of retinitis pigmentosa. Retinitis pigmentosa has approximately 200,000 patients. This is about 10% or 20,000 between the US and EU. It's pretty devastating, though, because the boys who get this have night blindness in their first decade of life. This disease then continues to progress. You get constriction of the visual field, so you get what's basically tunnel vision. And by the age of 40 or 45, due to vitreal hemorrhage and a variety of other things, most patients are blind. So this is a little tougher disease to treat because there's a slow progression. It is about a year behind the two that we just talked about. And as you can see, we've working on our preclinical now. We will have our vector constructs selected later this year, and then we will go actually into the clinic probably in 2016, assuming that the tox work in 2015 goes well. The visual constriction, a little slow. Okay, so we have one program beyond ophthalmology. This is our alpha antitrypsin program. This is a much larger program. Between the US and Europe, there's about 120,000 patients. Essentially, AAT is the third most ubiquitous protein in the body. It's made in the liver. It's then transported into the lung, where it works on neutrophil elastase. If you do not have this protein, you basically get lung degradation. It clinically looks like COPD. If you're not a smoker, it's often misdiagnosed or not diagnosed at all because physicians haven't really heard that much about it. If you're a smoker, most physicians assume that's your problem and it doesn't get diagnosed. There is some plasma-derived augmentation therapy out there, so the diagnosis rate is improving, and that actually may give us some help when we're thinking about reimbursement, which has been the big topic of this meeting for at least 2 days. We have worked on this in a variety of different ways, both with IM injections as well as another type of delivery. And what we have seen is that there is a linear dose response curve. And we also have seen expression, and this is, we say 2 years, it's really been 3 now with a single dose. This actually has been driving our manufacturing system, which we'll talk about in a little bit here. And this program will go into phase 2B also in 2015. Okay, so this is our team. Many of you may know Sue Washer. She's been in this business for probably the last 10 or 12 years, so you may have run across her. This is the rest of our team. So we do have our own manufacturing platform, which we've devised and developed ourselves. We just couldn't seem to make the other ones work, so we've been working on this for about the last 6 or 7 years. We found that it's called the herpes-assisted viral expansion platform. So we use the herpes virus to help us with our replication because, as you know, AAV is replication incompetent. We get excellent yield, 25 to 30 fold increase in potency. It's very simple to work on, quick to the clinic. Making the vectors does not take us very long at all. The characterization is there. We have over 30 qualified assays with our manufacturing system done either at AGTC or with our partners, SAFC, who's our CMO, or by Reliance. The scale is there. This is suspension culture. This is a completely industrialized process and drives our complete scale up. We are already at commercial stage for our ophthalmology programs. This one needs huge amounts of protein or vector genomes per cell and is driving significant scale up for us and makes our platform quite robust and I think one of the leading ones in the industry right now. It is reproducible. We've transferred it to SAFC. We also have a partnership with Genzyme around, or we did have a partnership with Genzyme around wet AMD. That is our manufacturing system. So it's transferable and scalable. And lastly, it has been reviewed by the FDA and the EMA and is approved for using human clinical trials. And as far as I know, that is the only manufacturing system that's been reviewed by both agencies. As I said earlier, we have over 100 patents. We continue to in-license from a variety of different academic institutions, and we have completely covered our manufacturing system with IP. The company did go public earlier this year in March, and then we did a follow-on in July. So we have about $100 million in cash. Our cash burn our monthly burn is about to go up as we're moving into the clinic. and some key milestones for us. So this year, Genzyme has presented their wet AMD phase 1/2 data. It was presented earlier in September at the Retina meeting. There's another presentation in October. So far, that's looked pretty good. I think they treated 19 patients, very safe. I think there were 4 or 5 that actually had some improvement. So it's pretty good for a phase 1. We've been very excited about that. We will file our IND for XLRS. We have our amendment for the alpha antitrypsin program, which I said will go to phase 2B in 2015. And then we are also working on a wet AMD program. This is new for us, and we think we will have some target ID for that moving forward by the end of the year. And then in 2015, the IND for Achromatopsia, a phase 1/2 data for XLRS and Achromatopsia. As I said, it's only a 12-week readout, phase 2B for the AAT, and then proof of concept for our wet AMD program. So again, really stepping out for us, really moving forward. It's a kind of a nice time to be at AGTC. So thanks very much.